All right, yeah, thank you, Akun, for that introduction. uh, so I am Sonal, I'm a postdoc in Wenyang's lab at UCL, and I'm going to be talking about a novel metabolic pathway that we found in the budding yeast, Saccharomyces cerevisiae, that deals with hydrogen sulfide. So I understand yeast metabolism is probably of niche interest here, so I do want to point out that what really excited us about the project was actually hydrogen sulfide, because it's a very interesting metabolite in itself, because the communal metabolite. Most cell types will produce hydrogen sulfide either by degradation of organic sulfur compounds or by reducing inorganic sulfates found in the environment. And once they produce that, the hydrogen sulfide gas can cross membranes without any assistance. They don't need transporters. And because the reduced sulfur is a very reactive species, it can produce a wide range of cellular responses. So high concentrations of sulfide are famously toxic for mitochondrial respiration, whereas at intermediate concentrations, you can get various positive effects, such as improving lifespan in yeast cells or protecting bacteria against antibiotic stress. So hydrogen sulfide is a pretty interesting and quite potent mediator of cell-cell interactions, it would appear. So with that in mind, what I'm going to do today is I'm going to introduce an unexpected observation we made in yeast metabolism and how that led us to realize that sulfide can mediate cell-cell interactions in budding yeast, and revealed a novel pathway of sulfide metabolism, and then finally led us to ask, what does this pathway really do in the cell? So yeast are one of the few lucky species who can actually convert inorganic sulfate into organic sulfur forms that all cell types need. And they do this by a series of enzymatic reactions that eventually lead to the reduction of the sulfur atom, so that hydrogen sulfide is produced and then combined with the organic substrate to produce the first organosulfur. Organosulfurs are all interconvertible in the cell, so once they have one, they can make all the others they need. So if you believe this pathway, which people have for very many years, if you delete this gene here, the yeast should no longer be able to synthesize their organic sulfurs and should therefore not be able to grow on inorganic sulfate unassisted. However, what we found was that MET17 deletion mutants could in fact survive on sustained growth on sulfate, and not just grow to saturation. So we're looking at turbidity of liquid cultures here. Not just grow to saturation, but if we keep diluting them, they would keep regrowing to high densities. So this was really surprising, because MET17 was classified as an organosulfur auxotroph as early as 1975. And it has been one of the background mutations in the yeast deletion library, which is a widely used resource in yeast genetics since 2002. So why would this get missed? Well, it appears that MET17's growth on sulfates is highly density dependent. So here, what I've done is I'm again tracking growth as turbidity, and I've started cultures at different initial cell density. And in each density, I have 3 technical replicates. We can sort of segregate the curves we get into 3 categories. At high density, growth is highly deterministic, so all 3 curves collapse into one. At low densities, you hardly see any growth in the cultures. And then there's this intermediate area, which is quite interesting, where you see the asterisk mark, where 3 identical cultures diverge in their dynamics. So we have stochastic lag times before the cultures appear to take off. Right, so this makes us ask, how do microbes count themselves? They usually release a molecule and also detect the same molecule. Let's call this molecule the growth mediator. We did a few preliminary checks, and then we got reminded that MET17 is perturbed exactly at the step where hydrogen sulfide is produced in high amounts. So could that be doing all this shady business? We first checked whether the growth mediator was a gas simply by parafilming our tube. So just added an extra level of sealing to our tube so that gas exchange was lower in the cultures. And we did in fact see that the same curves as before, different densities, 3 cultures at each density. If we seal them extra, even the low density cultures could now grow, albeit with quite some stochasticity still. We could also do another check where we put these different densities in the same multi-well plate, so they're sharing the same headspace now, the same air. And again, all densities can grow, and you would notice that there's much less stochasticity here. So it would appear that the differential concentrations that the cells are experiencing are giving rise to this and the stochastic growth we observed before. So we were curious whether yeast can really consume something like hydrogen sulfide from their vicinity, because hydrogen sulfide in acidic conditions should be protonated and mostly gaseous, and yeast cultures are pretty acidic. So, but this experiment would indicate that there is some amount of exchange that can take place at least. So what we did was we generated a sulfide consumer strain. So we deleted a gene which was upstream of hydrogen sulfide production, so that if now this strain has sulfide available, it should be able to just carry on organosulfur synthesis. As you'd expect, this strain, when growing alone in a multi-well plate, so these are all on sulfates, no external organosulfurs provided, they can't really grow very much independent of density. However, if they're sharing headspace with Met17 minus, then they can now grow quite a bit. What is also interesting here is that Met17 minus seem to slow down in the presence of these other consumers. So when the consumer to producer ratio has changed in the plates, growth has slowed down. So this tells us two things. One is that the volatile mediator is likely a sulfur-containing compound, and that the amount of it is determining when Met17 can grow and when not. Right, so to show that this is really hydrogen sulfide, one evidence we had was that lead acetate paper placed in these multi-well plates would turn black. It's a classical sulfide detection test. But we could also just add a sulfide donor, which is sodium sulfide, that releases hydrogen sulfide in acidic conditions. And if we added different concentrations, we see exactly what we'd expect. So again, we're starting with low density cultures. Even when we don't add any sulfide, one of them grew because stochasticity. And then we add some amount of sulfide, and now all three are growing, and there is some bit of stochasticity in the dynamics. You add a little bit more, and now the growth is almost deterministic. You can't distinguish between the three curves. And you can keep increasing it, and eventually you'll reach the spot where the yeast don't like it anymore and can't, in fact, grow. So this brings us to three sets of questions that we can ask. One is that, what is this pathway independent of Met17 that can actually assimilate sulfide in Cerevisiae? How does this pathway respond to sulfide, and what is the relevance of this backup pathway in wild type yeast, so when Met17 is functional? Right, so let's start with the first one. So yeast could not bypass the requirement for Met2, which provides the other organic substrate that's required for Met17's reaction. So we figured that whatever is bypassing Met17's requirement is going to do either the same reaction or something slightly similar. So what we proposed is we can find candidate genes by sequence similarity and then send them through this fairly straightforward screen, where we've seen that Met17 show density-dependent growth on sulfates. Gene X minus should not have any defect on growth on sulfates. But when you delete both genes, the cell should no longer be able to grow on sulfates at all. We collected a list of candidates, some from literature and some from sequence similarity, and we luckily stumbled upon the one gene that does meet the requirements of our screen. So as I mentioned before, you Met17 density dependent, delete this locus, and growth is independent of density, all cultures can grow. Delete both Met17 and that locus, and you don't see any growth at all. So this was pretty straightforward enough that two other groups discovered it at the same time. And one of them kindly named the gene so that I don't have to keep pronouncing that atrocious locus name for the rest of my talk. So it will be called HSU1 from now on, which stands for hydrogen sulfide utilizing. And the other groups also showed that if they purified the protein, it could carry out the same biochemical function as Met17. Right, so this brings us to the next question: how does this pathway respond to sulfide? Which is to ask what can give rise to this stochastic growth pattern that we observed before. So we hypothesized two mechanisms that could give rise to it. One was that all cells can, in fact, utilize the sulfide. So I'm only talking about Met17 minus cells here. So we're assuming Met17 is not available to them. But Even so, all cells can utilize hydrogen sulfide, but they can only do it in a low affinity manner when they rely on HSU1. So it takes a while before a cell accumulates enough organosulfurs before it can divide, and that can give rise to variable lag times. Alternatively, we could imagine a scenario where the cells actually need to be switched on first. So you could say transcriptional regulation or something that needs to be turned on first in a sulfide-dependent manner. And only when that is available can the cells then start to grow on that sulfide. So we could either have a low affinity pathway or the cell state switch hypothesis. Alex, who will be giving a talk tomorrow, helped me set up this model and run some stochastic simulations where the probability of either the switching or the cell birth is based on, has a monod dependence on the sulfide concentrations. And we could in fact see that in both conditions we could generate curves which have these stochastic lag times. So these are not with measured parameters. We just wanted to explain to ourselves whether these mechanisms can work to give rise to the curves we are seeing. So we could test these options a bit. So we could ask if there is cell state switch by doing the following experiment. We could take cells which have now started to grow, presumably have switched on now, and then redilute them at these different densities. And the idea would be that if you do see all cells can now grow, the switch was sufficient to explain the stochastic growth. And if you still see density dependent, there's at least no persistent switch that these cells have. If we do this experiment, we do see that this density dependence still exists. So it doesn't appear to be that there is a persistent cell state switch, and most likely there is the low sulfide affinity of HSU1 is giving rise to those stochastic curves, which is also something the groups that did biochemistry on it observed, that the affinity, the reaction is much less productive than at 17s. Right, so moving on to the last bit, what is the relevance of this pathway in wild type yeast? So is this just a backup pathway in yeast? Well, so the one thing was, you could wonder whether all this that I'm showing you is highly strain dependent, because there is quite a bit of strain differences in Cerevisiae. But the data I showed you was from a vineyard isolate RM11, but we also saw the same phenotype in the commonly used lab strain S288C. However, the requirement for the gene in the two strains is highly different. So you cannot delete the gene in RM11 because the cells don't survive, whereas in S288C, you hardly see a growth difference when the gene is deleted. So clearly, at least in a strain-dependent manner, the gene is doing something rather relevant. One of the other groups also did some phylogenetics analysis and found that the gene predates this genome duplication event that happened in Cerevisiae, and also it exists in a few genera apart from Saccharomyces. And then finally, hydrogen sulfide is not an unusual metabolite for cells to encounter, because it is an intermediate which doesn't accumulate often, but under certain conditions, such as nutrient stresses, nitrogen or glucose starvation, wild-type yeast can overproduce sulfide. So given all that in mind, we hypothesized one of two functions for this new gene. One was that the pathway maybe cleans up hydrogen sulfide when local concentrations get too high. If you remember a curve I showed you before, if I added too much sulfide, the cells were no longer able to grow. So it does appear that hydrogen sulfide can get to toxic levels for the cell, and this pathway might be cleaning it up. Alternatively, it might be a response to sulfur starvation. So to enhance sulfur uptake, make sure that nothing leaks out of the cell. And to discern between these two pathways, we would basically tag HSU1 with GFP, see under what conditions it's expressed, and then do competition experiments where we compete the deletion mutant with wild type and see under what condition does the wild type yeast have fitness advantage. This can of course only be done in S288C because in RM11 I can't delete the gene. Right, so the first option didn't really help us choose between the hypotheses, because it seems the gene is overexpressed both in high sulfide exposure as well as sulfur starvation. There's not much you see in exponential phase itself. On top of that, what we also noticed is that if you look at late stationary phase cells, it's a little difficult to see. I've highlighted them with arrows, but there are these puncta that where the protein also concentrates, and these are likely stress granules, which are also a cellular response to nutrient stress. So this basically sets up three conditions which we should test in our competition assays: high sulfide, sulfur starvation, and stationary phase. Fair enough, we started to do that, except we realized that the gene already has a pretty heavy disadvantage in the exponential phase. So what we're doing here is we mix HSU1 minus with wild type in about a 1-to-1 ratio, grow them under in just minimal media. And each of the genotype is expressing a distinct fluorescence protein, and we are counting cells at different time points using flow cytometry. And you see that the ratio steeply declines. So HSU1 minus steeply declines in number as compared to wild type. If you look at the turbidity in these cultures, and here I have 5 graphs that you cannot distinguish between, the 3 here, which are just replicates of these mixed populations, as well as monocultures of the 2 genotypes that I've mixed. And you don't see any difference between the growth profile and turbidity. So it doesn't appear that in isolation HSU1 has any growth defect. However, in the mix scenario, you can see this pretty distinct decline. So right, I have basically raised more questions than I have answered at this point. So it seems like a good time to wrap up the talk. So just to give you an outlook, I have shown you that this HSU1 can replace MET17's function in its absence. I have shown you that the stochastic growth of MET17 likely results from a low affinity for hydrogen sulfide. And then HSU1 is playing a non-redundant role. We just don't yet know exactly what it is, and it seems to be there throughout. So it starts from exponential phase, stationary phase, different nutrient stresses. So what I haven't shown you and would eventually want to is the competition assays done properly in these different conditions to see what more we can learn about it. We can also learn something from the fact that it has this different essentiality in the 2 strains. We can try to identify the other gene loci that determine why it is essential in one strain or the other, and that may give us some idea of the functional networks of the gene. And finally, I did want to mention that the thing that this whole project did for us that really made us think about the gases in our cultures, which we almost always ignored. So if I can be so presumptuous to give anyone advice in this room, I would say do pay attention to the air in your cultures if you see phenomena you can't explain. And thank you so much for your attention. I'll leave you with the acknowledgement slide here.
Thanks a lot for that. We have time for a few questions. Maybe I got 2 questions actually.
Yes.
So there's a suggestion of phenotypic basically heterogeneity. So do you see any heterogeneity in your HSU1 gene expression?
No, not really. We thought that the heterogeneity might come from expression, but we don't really observe anything.
And the other one is more like a comment that the hydrogen sulfide is also implicated in oscillation synchronization in thermostat cultures.
Yeah.
Did you think about that, or did you look at that in HSU mutant?
Yeah, so I mean, at this point, actually, to be honest, I'm not very sure whether it's actually doing anything with the sulfide itself, or if that's just a condition that arises in this unnatural MET17 deletion condition. But we haven't really looked at anything of that nature. What I do see is that hydrogen sulfide toxicity has not changed in HSU1 minus. So it's not that they become more sensitive to hydrogen sulfide. But it might be interesting to see in co-culture conditions whether hydrogen sulfide released by wild type cells is somehow affecting the growth of it. So if we could do those assays also in a multi-well rate setup.
Thank you.
Yeah, sorry.
I really enjoyed that. In your simulations, you only had them up very fleetingly. It looked like in the 2 models, you actually did see one of the scenario that you found out that did look a bit more like your experimental work.
Yeah.
Is that true, or is this
So, the parameters are really arbitrary. So I don't know how much to rely on that, but I do find it's easier to recreate the curves that look like the data from the low affinity version rather than the cell state switching version. It is, of course, possible there's a mix of both as well, right? So I mean, also because I've looked at gene expression at this point in time, I do know that the gene is present early on. So it's not that the gene only comes up once enough sulfide accumulates. But it could still be a mix of both that is doing it, yeah.
If not, let's thank Sonal again.
So much.